morning, we are Group 1. Today, we will be talking about Lesson 16, Introduction to Analytical Separation. Previously, we presented about mass spectroscopy. We learned that it provides rich elemental information, which is an important asset to interpret complex mixture components and unknown compounds. Today, we will explore the different analytical separation methods, techniques, as well as the microanalytical systems. But first, here is our learning objectives. To describe analytical separation, to enumerate the different separation methods and techniques, and to understand the advantage of microanalytical system. And here is our conceptual framework. What is separation? The word itself means the act of separating or the condition of being separated. In analytical chemistry, separation process is a method that converts a mixture or solution of chemical substances into two or more distinct product mixtures. It exploits differences in chemical properties or physical properties between the constituents of a mixture. What is the purpose of separation? First is to remove unwanted particles and impurities. Second, to obtain important substances. Some substances are more important than others in a mixture. Therefore, this is need to be obtained by separation of substances. And third, to obtain pure substances. A mixture is actually made up of pure substances only, but they get mixed up with each other. Sometimes, individual pure substances are more important than the mixture. Separation process is needed to separate the useful components from non-useful or harmful one, because it may spoil the useful component in the mixture. Two general reasons for performing separation of mixtures are: first, the mixture may contain some substances that should be isolated from the rest of the mixture. And second is to alter the composition of a sample so that one or more of the components can be analyzed. There are two methods of separation: the physical separation method and the chemical separation method. Physical separation methods are based on the boiling point of the substance in the mixture. These are properties that you can observe and measure without changing. Under these methods are filtration and distillation. Filtration is the simplest form of separation, which separates the components of a mixture by using a funnel lined with a filter paper to retain solids while letting the liquid through. While distillation is a separation technique used to separate components of a liquid mixture by a process of heating and cooling. Now, the chemical separation method. It is based on chemical interaction between the mixture's components. Naturally, these interactions are based on the chemical properties of the materials to be separated. Chemical separation methods include extraction and chromatography. In extraction, a solute is extracted from its solvent. Example of extraction process is the separation of DNA from proteins of biological sample. On the other hand, chromatography is a separation technique based on how the different components in a mixture have different affinity for the stationary and mobile phase.
Now let us know what is separation techniques. Separation techniques are those techniques that are used to separate mixtures into its constituent elements and compounds. Recall that a mixture contains elements or compounds which are not chemically combined together. By separating the constituents of the mixtures, we are able to find out the properties of the known or unknown substances from mixtures, and possibly use them for the production of useful substances such as medicine. Next is the classification of separation methods. All separation methods of homogeneous mixtures were divided into 5 groups. First group is that methods that are based on the formation of new phases by substances to be separated. Second group, methods that are based on differences in the interphase distribution processes. Chromatographic methods being singled out as a separate group. Third is that membrane methods that are based on the induced transfer of substances from one phase into another one across a third phase, which separates the two. Fourth, separation methods within a single phase that are based on velocity and direction, differences in spatial displacement of particles of substances to be separated within fluid phase under the action of various fields. And fifth, combined methods combining previously listed methods. Next, we'll proceed to the most common separation techniques. I have here the 5 most common separation techniques, which are the filtration, distillation, crystallization, chromatography, and centrifugation. First is the filtration. Filtration is a separation method used to separate out pure substances in mixtures comprised of particles, some of which are large enough in size to be captured with a porous material. Particle size can vary considerably, given the type of mixture. For instance, stream water is a mixture that contains naturally occurring biological organisms like bacteria, viruses, and protozoans. Next is the distillation. Distillation is an effective method to separate mixtures comprised of 2 or more pure liquids. Distillation is a purification process where the components of a liquid mixture are vaporized and then condensed and isolated. Third is the crystallization. Crystallization is a technique used for the purification of substances, a separation technique to separate solids from a solution. Crystallization can be defined as the process through which the atoms or molecules of a substance arrange themselves in a well-defined three-dimensional lattice and consequently minimize the overall energy of the system. When a substance is subjected to crystallization, its atom or molecules bind together through well-defined angles. Next is the chromatography. Chromatography is the separation of a mixture by passing it in solution or suspension or as a vapor through a medium in which the components move at different rates. Thin layer chromatography is a special type of chromatography which is used for separating and identifying mixtures that can be colored, especially pigments. Lastly is the centrifugation. Centrifugation is a technique used for the separation of particles from a solution according to their size, shape, density, viscosity of the medium, and rotor speed. The particles are suspended in a liquid medium and placed in a centrifuge tube. The tube is then placed in a rotor and spin at a defined speed.
Now let's proceed to microanalytical system. Micro-total analysis or microanalytical system, better known as lab-on-a-chip devices, are miniature devices with integrated functionalities including sample loading, sample treatment, separation, and detection. It is usually done in laboratory analysis such as DNA sequencing or biochemical detection. The basic idea of microanalysis system is to replace those laboratory liquid management tasks with a liquid handling circuit device and reduce an entire laboratory room down to one chip. And this is why microanalytical system is also frequently referred to as lab-on-chip. Microanalytical system is based on microfluids, a multidisciplinary field that involves precise control of fluids at a sub-millimeter scale. Now, what does it mean when we say microfluidics? It is the science and technology of systems that process or manipulate small amounts of fluids, using channels with dimensions of tens to hundreds of micrometers. A microfluidic device handles liquid, just it exploits the physical and chemical properties of liquid and gases at a microscale. A microfluidic devices have a micrometer scale trench structure in which the liquid flows. Therefore, a very small volume of liquid can be handled and used for chemical reactions in these devices. These characteristics of microfluidic device make them best suited for analyzing biological and medical samples that are difficult to collect in a large volume for sample analysis. The major advantages of microfluidic devices are low experiment to cost ratio and fast response time. This microfluidic chip is a pattern of microchannels which is molded or engraved. And this network of microchannels incorporated into the microfluidic chip is linked to the microenvironment by several holes of different dimensions hollowed out through the chip. Now, it is through these pathways that fluids are injected into and evacuated from the chip. Fluids, as what you can see, are directed, mixed, separated, or manipulated to attain multiplexing, automation, and high-throughput systems. Microfluidic devices offer innumerable applications in the field of science and technology, and their scope is not limited to single field and now have applications in various fields like in biomedical, as analytical gradients, separators, microdiluters, gel structures, droplets, painting cells, and devices. And this type of system can be used for detection of specific proteins in large number of samples, antibiotic resistance, and etc. It can also be applied in the field of energy, in the form of fuel cells, wherein all the systems such as fluid delivery, removal, and others is confined to the microfluidic channel only. Microfluidics has also been observed in the field of environmental assessment, as multiple processes such as pre-treatment, pre-concentration, separation, and detection are incorporated at the same platform, which used for trace analysis of materials as less risk of contamination.